Hello, my name is Jenny Ricker. I am here to verbally discuss the article written by Giorgio Bernardi about genomic code and the pervasive, and I believe it was molding of both the chromatin model as well as the that would be the non-coding DNA mystery. So, the article begins discussing how the genomic model as well as the non-coding DNA were not very well respected in terms of importance of function in the genetic world. They were looked down upon as being fairly useless. So as time goes on, there were techniques that were created in which to divide and split DNA to further research and get to know more about these two particular subjects. So there was one technique, which was chromatinography. That was when you split the native DNA with the collapsed DNA for investigation. Then the second is ultra transfiguration. Sorry, that one's hard for me to pronounce. That there is a great example. So it works with high frequencies. That's how it's able to divide DNA. They had a successful example of applying it to a mammalian, and they were able to divide it 3 ways. So these techniques led to further research, and through further research, specifically through heat chromatin maps, and then also very distant low-high loci maps, they were able to prove a relationship between the two isomers GG poor and GG rich. I love those names, by the way. Really funny. They correspond actually with 2 other pairs that are very important in genetics. I don't know why I'm holding this. Sorry. LAD and TAD. These also isophores located within the genome. What the conclusion of the article shows is that there is a huge association between the LADs and the TADs critical for the chromatin relationship with genomic modeling and also non-coding DNA. Genomic taboos are no longer present. They used to, I think at one point, refer to them as a beanbag. They're now highly regarded and understood as highly functional. Now, in my opinion, I'm very pro this. I think that any scientific advancement that could help with potentially disease or gene therapy to prevent disease, whatever gives us a wider range of material to work with to prevent or cure, I'm all for it until we find out something later where there might be a big drawback. I hope you enjoyed this. I thought this was a great article, and thank you. Bye.